Hey, hello everyone. I am JJ, your mentor or your friend or whatever you want to call me. Yes, so I hope that you're all having a very great day today. If you're not having a great day, then I wish that you have a beautiful day. Okay, so we have reached the climax. Actually, we haven't, but we are about to reach the climax. We are in the part 4 of apoptosis, and we now have so much knowledge about apoptosis. We know what are the basics of apoptosis. We also know whether there are some other process like apoptosis, and we know what are gas spaces. So gas spaces are heroes of our movie, which is named Extrinsic Pathway. The movie's name is literally very boring, I agree, but it is not that boring actually. So, yes, you can watch that movie in this lecture. But before that, if you haven't heard of gas spaces, you should definitely go and watch the previous video that I put. I will put a card over here somewhere here in the top right corner. You can click on that suggestion and so that you can watch what is Caspace. Okay, so yes, and for those who have already seen, I'll give a quick recap of Caspaces so that you can understand the movie better, okay, in a better way. And those who are thorough with gas spaces, you can just skip this portion. Yeah. Yes. So this particular thing that you see on the screen is a protein, okay, and caspases are protease. So protease are enzymes which breaks down proteins. Okay, but they are somewhat special. We will just read out what it stands for. Okay, so it is cysteine dependent aspartate specific protease. Cysteine dependent because they have cysteine amino acid in its catalytic unit, and so that's why it is cysteine dependent. It's dependent on cysteine. And then it's aspartate specific, so it is specific for aspartate. Aspartate is an amino acid, so it searches for aspartate and then cuts at the C terminal of aspartate. It is specific for aspartate. It just cannot go and cut any other amino acid. Okay, so that's about caspases, and as I told that they are responsible for breaking down proteins. So what proteins? So usually the target proteins are those which provides the structure to the cell. Okay, so if these proteins are cleaved, then the structure of the cell will be lost, and that's how apoptosis occurs, right? So that is the way how caspases actually work. So these caspases are not always active. They are found in the body in the inactive form, and they are known as procaspases. And for making them active, they need to undergo cleavage. So we will see how. So there are two different conditions for undergoing cleavage. This thing you see is procaspase. This is one procaspase. This is second procaspase. Okay. So what happens is that the first condition is it has to attach to an adapter protein. So adapter proteins are things which further attach to some receptors, okay, and that's how the pathway works. So the first condition is that it has to attach to an adapter protein, and then the second condition is that it has to be in close proximity with other such procaspases. So when these two conditions are fulfilled, like right now, these two conditions are fulfilled because they are attached to an adapter protein and also they are in close proximity. So they now can cleave each other at two different sites, that is site 1 and site 2, and we will be left with a large subunit and small subunit. The prodomains actually lives on the adapter protein itself, and then these large subunit and small subunit form a heterodimer. Okay, still they are sadly inactive. So for making them active, both of these heterodimers should come together and form a tetramer, and then they will be active. Tada! So when they are active, they go and cleave executioner caspases, which will cleave proteins that I mentioned earlier, that is target proteins. Okay, so these initiator caspases are for initiating and attaching to adapter proteins, whereas executioner caspases are for actually doing the work. So now we will get into the main topic, that is extrinsic pathway or death receptor pathway. I know it sounds like a horror movie, but it is not. Death receptor and all, right? It's not at all scary. Let's see that. Yeah. So first of all, we will start with the basics, that is about receptors and ligands. So there are some receptors which are present on the surface of the cell. They are known as extracellular receptors, and there are certain receptors which are present inside the cell. They are known as intracellular receptors. On these receptors, ligands comes and attaches, and that's how the process goes on. Okay, so here in the case of extrinsic pathway, there are certain extracellular receptors. You can see that here, right? And these receptors are known as Fas receptors. Okay, they are known as Fas receptors, and they are trimeric. They are one, two, three. So they are trimeric, and they are known as Fas receptors, and they are also known as death receptors. But why? Why death receptors? Because they have a death domain at its cytosolic end. This blue thing you see is a death domain. Okay, so that's why they are known as death receptors. But of course, for the pathway to work, there's a need of a ligand. So there are ligand, they are also trimeric, and they are carried by different kind of cells. Actually, they are usually carried by killer cells or TC lymphocytes, which are there for immunity. Okay, so these ligands are known as FAS-L, that is, the receptors are known as FAS receptors, so the ligands which binds to them are known as FAS-L, that is fatty acid synthetase ligand, and you have to learn the name. Okay, fatty acid synthetase ligand attaches to the fatty acid synthetase receptors. Okay, and these receptors and ligands belong to tumor necrosis factor family. This is a huge family of receptors, but mainly we are studying fast because they are the most studied ones. Okay, so now let's imagine that the ligands have attached to the receptors. Okay, and these receptors have death domain. So this thing you see is Fas receptor and ligand, and there is caspase. So I told that for caspase to get recruited into the pathway, they need some adapter protein. So this middle portion you see, this middle thing that is present, is known as adapter protein, and it is named FADD, which stands for Fas associated that domain. I will explain the name. That is, it is associated with Fas. Okay, it is associated with Fas with its death domain. So FADD also have a death domain. Okay, it has a death domain which attaches with the death domain of Fas receptors when ligands comes and attaches, and then it also has death effector domain. These death effector domain attaches with the death effector domain of caspases, which are found above the prodomains. Okay, so there is death effector domain that attaches with the death effector domain of FADD, or simply an adapter protein, and that's how they come in close proximity, and they now cleave each other and form active tetramers. So these caspases which are involved here are caspases 8 and 10. Okay, and all of this forms DISC. DISC stands for death inducing signaling complex. As the name suggests, they are the complex which induces death. Okay, so it starts from DD, that is the death domain of receptors, and it ends with the caspases. So that is what DISC is. Yes. So now we'll see what happens to the active caspases. Okay, so there are active initiator caspases 8 and 10 here. They will now cleave executioner caspases 3 or 7. Okay, these executioner caspases can now either cleave other executioner caspases, that is their friends, or they can cleave certain proteins which would lead to the DNA fragmentation, or they will cleave nuclear lamin proteins which forms the nuclear lamina and gives a structure to the nucleus, or they can cleave the cytoskeleton proteins which provides the structure to the cell. Okay, so all this will lead to the loss of structure of the cell, and at last it would result in apoptosis. So that's what we wanted, right? So now I want you to stop, breathe, and revise. I have made notes for you in these slides, so you need to go and revise that, and it will surely help you. So yes, do that, and after that we will come across the main villains of this particular pathway. Basically, there are two villains. First is C-flip proteins. So they are proteins which the cell produces at times. Okay. So C-flip proteins are the main villain. They actually fight with a hero, that is caspase. Okay, and caspase usually binds with FADD, right? So C-flip protein, that is a villain, binds with FADD instead of initiator procaspases, that is a hero. Let imagine that FADD is a heroine, so they both fight for a heroine, and sometimes civil protein wins, and that's when they that is the initiator procaspases are left out, and they won't actually lead to apoptosis because they are not attached to adaptoprotein. They need to attach to adaptoprotein for its working, right? So if it's not attached to FADD, of course they cannot lead apoptosis, right? So that's how it works. And the second thing is that sometimes cell produces decoy receptors. These decoy receptors fools the ligands which are present. The ligands think that they are the death receptors, but they are not. They are decoy receptors. They do not have a death domain inside the cytosolic end, or this in at the cytosolic end. Okay, so they just attach to the ligands that attach to the decoy receptors, but the process, that is the pathway, won't be initiated as there is no death domain. And that's how it works. That's how the villain does its job. So that's it about extrinsic pathway. I thank you from the bottom of my heart. So if you like my video, please like and share and subscribe and encourage me, because that's what you can do for me, right? So yes, thank you. Much love. Bye from JJ.